Glioblastoma is an incredibly aggressive human brain cancer, and scientists may have finally found an effective treatment. Before I begin, I just wanted to say, of course, I might get things wrong. The terminology used in the article somewhat confused me, but I tried my best using other sources to explain what it meant. I am by no means a professional. Hello, welcome back to my channel. If you have watched the last video, you probably have realised my voice has changed, and hopefully for the better. I got a new microphone. Um, Hopefully I sound better and clearer compared to last video. But anyways, at the time of the video being recorded, it is the 11th of March 2026, and the extremely interesting article, which is on glioblastoma, again, or GBM for short, which is brain cancer, I'll be covering today, isn't the newest. It was released on the 31st of December 2025. However, the findings and procedure of this study are extremely fascinating, so I thought, although it is an older article, it would definitely be worth sharing and going over, as the findings could lay the foundation of future brain cancer treatment. Going over really quickly why I find this such an important article is that glioblastoma, which I will be calling GBM from now on, is the most aggressive malignant primary tumor. This basically means it spreads quickly, they are hard to kill, and people who have these cancers typically have a low survival rate. And by primary, it means it isn't part of another cancer that broke off and later multiplied. In other words, metastasized. GBM is most often treated with surgery and radiotherapy, specifically ionizing radiation. And after that, the DNA alkylating agent temozolomide, or TMZ for short. The way TMZ works is it binds to the DNA in cells and it specifically interferes with cell growth and division, preventing the tumor from multiplying. Although this combination of the three provides some progression-free survival, there is no cure. With the median survival rate with standard care being 12 months. GBM is one of the most treatment-resistant human cancers, with progression and recurrence being one of the main features. During TMZ treatment, MGMT, or methylguanine methyltransferase, which is crucial for genome stability, repairing the naturally occurring mutagenic DNA lesion of O6-methylguanine back to guanine. Oh my god. MGMT promoter hypermethylation caused silencing of MGMT, which is the enzyme, which played an essential role in TMZ-initiated death of malignant tumor cells. However, approximately 80% of GBM patients experience recurrence at or near the original tumor site, so it grew back, pretty much. However, scientists found that a combination of TMZ in EDU, or 5-ethanol-2-deoxy... However, scientists found a combination of TMZ and EDU, or 5-ethanol-2-deoxyuridine, provided highly significant survival benefits and synergistic effects, which means the combined chemicals provide a larger positive effects, greater than the sum of each individual drug. Going on to how they thought of this combination, let's start with EDU and how it provides a positive effect. Researchers luckily found that EDU is incorporated into the human genome. It is recognized as damaged by the cell's repair system, and then it is removed by cutting out small pieces of the DNA, about 26 to 7 nucleotides long, which means that EDU keeps getting added back into the DNA and then removed again, and then added back and removed, creating a repetitive wasteful cycle as it removes more than is added back. This finding, combined with previous reports on toxicity of EDU in tissue culture, as well as its already known property of crossing the blood-brain barrier, suggested EDU might be effective against brain cancer. Now, onto the procedure where they found the effectiveness of the combination of both drugs. They began by establishing normal GBM tumors in female anthemic nude mice by inoculating, in other words, growing. Inoculation is the act of implanting a pathogen or other microbe or virus into a person or other organism. Basically growing it inside of the mice. By inoculating human GBM cells into their brain parenchyma, or simply into their brain. Following confirming the establishment of the tumors using bioluminescence imaging, or BLI for short, they randomized mice into 4 groups. Group 1 had a PBS control, It's phosphated with saline, so researchers can compare treated groups with this group. Group 2 had 200 milligrams per kilogram of EDU. Group 3 had 5 milligrams per kilogram of TMZ. And Group 4 is just Group 2 and 3 combined. It's 5 milligrams per kilogram of TMZ and 200 milligrams per kilogram of EDU, with the injections being administered for 3 consecutive days. The brains of the mice were then later harvested the following day. Wow. Yeah, I completely forgot about this part. That's kind of weird to think about. I actually used to have two mice, one after the other, not both at the same time. One of them died of cancer. Both of them died of cancer. It's a strange thing with mice or rats. They always die of cancer, it seems. But I digress. The brains were harvested about 18 hours after the last injection, so that they could stain them with a variety of chemicals such as hematoxylin and oosin, nuclear and EDU staining. These stains show that in a control group with PBS and also the 5 milligram per kilogram of TMZ group, EDU signal was not detected in either tumor or non-tumor cells. On the other hand, EDU signal was highly localized to the tumor cells in both groups of mice treated with 200 milligrams of EDU per kilogram, and the other group being 5 milligrams per kilogram TMZ and also 200 milligrams per kilogram of EDU. Quantification of EDU positive cells in tumor versus non-tumor cells found that approximately 83 times more EDU positive cells were in the 200 milligrams per kilogram EDU group and 70 times more in the EDU positive cells in 5 milligram per kilogram TMZ plus 200 milligram per kilogram EDU treated group. So both of them combined. The residual EDU signal shown in this image, that I'm going to put on the video if I don't forget, was localized mostly to the subventricular zone of the brain, known to harbor some proliferative cells. These findings confirm the highly targeted localization of EDU cells into tumor cells of an organism. However, EDU distribution in plasma and the brain has not yet been characterized. So the researchers performed a pharmacokinetic study with healthy female anthemic nude mice. What they did was they administered a single dose of 200 milligrams per kilogram of EDU. They collected plasma and brain samples from them at 6 different points in time, and they obtained the EDU concentration. They found that maximum plasma concentration of EDU was reached at 15 minutes at 73 micrograms per milliliter with a half-life of 10 minutes. However, in the brain, the maximum concentration of EDU in the brain at 15 minutes was 4.8 micrograms per milliliter with a half-life of 30 minutes. Together, these results support the hypothesis that EDU localizes into the tumor cells in the brain in an organism. They have covered so much more in this study that I just cannot go over because of the sheer amount of volume and studies in one article. I will, just like in the last video, link the study in the description if you want to check that out for yourself. There's so much. But anyway, the results on combining EDU with other chemotherapeutics against GBM patient tumor tissues reveal the transformative role EDU could play in alternative treatments for GBM. Future studies on the mechanism and therapeutic potential of these combinations will hopefully highlight the clinical utility of each in GBM. These combinations could be personalized and provide much-needed alternative therapies. Not therapists, therapies. Wow. Reading through the article, I felt like I was reading an ancient Chinese script. Jesus. And honestly, half this terminology I didn't even know existed up until I started making this video. I had to search up what half of it read, and none of it was familiar. None of it. I find it really hard to keep with the terminology, but I eventually got my head around it. Hopefully, I also did that for you guys. I hopefully made that easy to understand. Again, this article is a good foundation for future research into the combination of a variety of drugs to treat GBM. Makes you think, really, how close are we really to finding a cure for cancer? Although this is only into GBM and only on mice, GBM is one of the most aggressive brain cancers. Hopefully, which I doubt it does, This means that the less aggressive ones will be easier to treat, and we can get a fuller understanding of how cancers work and can be treated. Another question. How much will the 168,000 people die from cancer per year in the UK on average, 406 people per day, how much will that number be reduced? Just take a second to think about how much 460 people is. I will put an image on the screen of like around 500 people in an audience. It's an insanely big number. How much will this be lowered? I think it's going to shoot down, but after all, we all think differently, and pessimists exist. Now, I just want to leave you to something to think about. With all the greed and malevolence of people, should we be making a brain cancer treatment only for humans? I think everyone who has had animals or a family has gone or has experienced somebody or yourself having cancer. Maybe this is more directed towards animals. If they have cancer, there's no cheap option. They're good. I have had so many animals that died of cancer, and if they do have cancer, the vet is so expensive. You're kind of cooked. The dog's cooked. But anyway, yeah. If we kill our planet, do only we deserve cancer treatment? There doesn't seem to be any viable option or cheap option for animal owners, which seems completely wrong to me. But I think the philosophical yap. I hope this video provided some insight into current brain cancer research, and I managed to explain it in an understandable and good way. And I hope you learned something new. Thank you for watching, and I will see you later. O kurwa.